Hello boys and girls, this is Mr. So. Welcome back for another lesson of biology. Today we are going through lesson 25 of chapter 14 about the apical meristem. Before you start watching the video, remember to like and subscribe my channel and also share the video to your classmates. Let's start. After the seed germination, seedling will be formed a very small plant, and then this very small seedling plant, it will grow bigger. Now the question is, where does growth and development take place in the very small seedling plant? Growth actually takes place in meristems. It is a group of undifferentiated cells, very small and simple, and they are able to do mitotic cell division throughout their lives to increase the cell number. There are two types of meristem. The first type, we call them the apical meristem, 顶端, which you can find it at the shoot tip at the very top and the root tip at the very bottom of the plant. These type of apical meristems helps the plant to increase in height, to grow taller and deeper. The other type of meristem, we call them the lateral meristem, which in Chinese 脏. It means for those meristem, we call them lateral meristem. They helps the plant to grow laterally, to grow in the width. Let's focus on the apical meristem first. Apical meristem can be found at the tip of the shoot and the tip of the root. It helps to increase in the length and the height. We call this primary growth. This is the very first task a plant has to do when they first germinate. That is to increase in the height or increase in the length, primary growth. Let's take a look at the structure of the root tip. At the very ending part, you can see this is actually the root cap. And right behind the root cap, you have a bunch of very tiny small cells. And this is a group of cells, we call them the apical meristem, to help the plant to increase in the length. So what is the function of the root cap? It is actually to protect the tip of the root. So let's write it down together. Protect the root tip. That means to protect the apical meristem. You can find the apical meristem right behind this root tip. The roots can be also divided into different regions. The regions at the very bottom near the root cap with the apical meristem in between, we call them the region of cell division because apical meristem do a lot of cell division. And then moving up, you have a region with the cells a lot longer in the length. In here, the cells, they are very long in general. This is the region of elongations for the cells to increase in the length. And for the last region at the higher part is a region of differentiation. The cells now, they are no longer just a simple cell, not a longer cell, but instead they have differentiated into different functions, such as you can find the xylem and root hair in this region. Xylem in the middle, and you can find root hair at the side. This is a region of differentiation. Let's take a look at the region of cell division first. It is right behind the root cap or containing the apical meristem. Many small tiny cells. And the apical meristem produces new cells by the mitotic cell division. The diagram below shows you the root tip. Now, can you find out where is the root cap? Give you 3 seconds to find it out. The root cap is located in here. This is the root cap. It's containing a lot of dead cells. Where do you find the apical meristem? You can find it in here. Alright, these are very tiny small cells, and they are doing rapid mitotic cell division to increase in the cell number. For the cells in the region of cell division, they are very tiny, very small, with a very dense cytoplasm. Moving up a little bit, this is the region of elongation. And for the cells, in the region of elongations, all of the cells come from the region of cell division. How can a very tiny cells from the region of cell division goes to the elongation region and elongate themselves, increasing the length? That is by taking in water by osmosis. After the cell is filled with water, the cell wall will be stretched and then the cell elongates, increasing the length. After the cells elongate, become longer in the length, the cells which elongates, they will push the apical meristem downwards, and this is how the root they will grow further down into the soil. For the cells being after elongated, become longer in the length, the cells will later differentiate into some specialized cells for different functions, such as the cells on the side as the root hair. Or you can find the cells at the center of the root, such as the transporting vascular tissues. You can find the xylem vessels for water transport, or sieve tips for the nutrient transport, such as the sucrose. The slide in this page shows you a table and a summary of the region of cell division and the region of elongation, as well as the region of differentiation and their details. You can pause your video and take a look at it for a while. Let's do a questions to round up what we have learned so far. The diagram below shows a longitudinal section of a root tube. Using the letters in the diagram, can you identify the region of cell division? And also the region of differentiation in the root tube. Where is the cell division region? It should be on Y. What about the region of differentiation? You can find it at the very top where you can find the root hair. It should be W. And now, can you name the group of cells? A group of cells that produce new cells in the region of cell division. It should be the apical meristem. Last question, what is that? At the very top of the root tube, it is actually a layer of dead cells. And this layer, we call them the root cap. And the function of it is it protects the root tips. Where you can find the apical meristem. Please go to your textbook page 14 to finish the MCQ 1 and MCQ 3. The root of the plant is quite long, so how can you find out the elongation region? Let's take a look. We can find out the region of elongation using a scientific method. Take out a marker pen, and then starting from the tube, try to mark down some lines across the root at 1 mm interval, such as take out a marker pen and then mark out a line 1 mm interval. After that, allow the seedling to grow for 1 to 2 days. After the root grow out, measure again the length between the adjacent line. The region which has the greatest increase in the length is the region of elongation. Or you can say the spacing between the lines should have the greatest in length in the region of elongation. Let's go back to the classwork. Can you design an experiment to find out which part of the root is the region of elongation? Copying from what we have just done, we should start from the tube and mark down some lines across the root at 1 mm interval. Let's write down together. Starting from root tip, mark some lines across the root at 1 mm interval. So you get the first mark, and then you allow the root or the seedling to grow for 1 to 2 days. You get a second mark. Afterwards, you will start to measure the length between adjacent lines. You get the third mark. The very last mark. The spacing or the region with greatest increase in length or the spacing between lines with greatest increase is the region of elongation. You get the last mark. Besides finding apical meristem in the root tip, you can also find it in the shoot tip. Those cells who are very tiny at the very top of the shoot tip, they are the apical meristem. This is the shoot tip, and you can find the apical meristem in here. Similar as the root tip, you can find the region of differentiation at the very top of the shoot tip, and then moving down, you can find the elongation region, and then the region of differentiation, where you can find the xylem and phloem and etc. different types of tissues with different functions. That's it for the lesson about the apical meristem on the shoot tip and also the root tip. I hope you liked the video. I will see you in the next lesson. Bye bye.